Thank you so much again. It's great to be here. I'm going to be talking on something that is the center of the research at my laboratory at North Carolina State University, and that is how to understand biological networks. And biological networks are something very dear to me, but before we move forward with that, I had to say this. I'm helping a friend to start in a startup, so I had to disclose this conflict of interest. With that out of the way, I would like to talk about cellular transitions. And living systems have an amazing capacity to undergo transitions. Just imagine the first time that you understood that a caterpillar can turn into a butterfly. This usually inspires poetry, it's even in sacred books. But as the biochemist that I am, when we look at these transitions, what we look at is actually protein networks, where we have a network of proteins in one configuration at time one, and at some future state it changes, usually by acquiring new proteins or rewiring its interactions. And in my laboratory, we are really interested in how can we directly visualize and measure these protein networks. This is a non-trivial problem. We see networks in many different places, but I will argue that most of them are theoretical constructs. We use a model organism called batting yeast, which has a lot of networks that have been really well characterized. For example, it doesn't matter that you don't know these proteins, but if you look at this little network, it basically has a protein that then triggers a cascade that is going to activate these two proteins, CLIP1 and CLIP2. Whenever you activate these proteins, you enter a state called the high CDK1 state, because these proteins activate a kinase called CDK1. And when CDK1 is active, the cells want to divide. So this state brings us to mitosis. So when cells divide, they are in a high CDK1 state. This network is active. However, as you may imagine, sometimes the cells don't want to divide. So what do they do to this network? They put an inhibitor on top. This inhibitor is called V5, and when V5 is active, the whole network shuts down. This state is a low CDK1 activity state, and it gives us states, for example, like mating. Cells can mate and terminally differentiate when the network is in this position. Now, sometimes the network needs to include also environmental and stress signals. And when that happens, there is a gigantic reconfiguration. You have transcription factors that get activated in response to stress. They trigger other proteins. These proteins wake up other proteins that were previously deactivated, like CLIP5. And overall, they start to produce other transcription factors so that they finally get to produce more of this protein, CLIP1. And when we get to this point, now the network is again in a high CDK1 state. Now, this configuration is really interesting because it gives us cell division, but it's not mitosis, it's meiosis, which is the basis of sexual reproduction on the planet. That's how fungal organisms make spores, and that's how we make egg cells and spermatozoids. But sometimes the cells really don't want to do absolutely anything because the conditions might be too harsh. And in that case, they can still produce another transcription factor called XBP1. And when XBP1 is produced, the whole network can shut down. We enter a new period of low CDK1 activity, and this one is also special because it allows the cells to remain dormant. And what that means is that the cells now can survive antibiotics, for example, if they are microorganisms, or they can remain dormant, for example, like the stem cells in our tissues. Now, all of these networks, believe it or not, have been studied in isolation. So we have specific laboratories that look at mating or dormancy or meiosis or mitosis. It's very hard for us to actually look at the transitions when one network is becoming a different one or is transitioning into a different configuration. And that occurs because there's a problem with how we study these networks. This doesn't happen in a test tube. These cells are actually changing shape. For example, the transition from mitosis into mating takes a couple of minutes. The transition from an arrest into meiosis can take a couple of hours. And finally, the transition into dormancy can take a couple of days. And during all these different transitions, the cells are changing shape dramatically. So if you want to study the networks, you had to face two questions. The first one is, how do you follow the cells while they are undergoing all of these transitions. And the second one is the networks are composed of multiple elements. So how are you going to track every single one of them? That's a problem that we're going to tackle in this talk. So in the first part of the talk, I would like to talk about how to track the cells during these transitions. And in my laboratory, we also use microfluidic assays. So this is an assay in which we're going to try to trigger all the transitions in the full life cycle of batting yeast. So we start with a cell that is going to be forced to make a decision either to make spores or not to make spores. After that, we found a drug cocktail that allows us to specifically select the cells that are making spores. We want those cells because those cells, upon exposure to rich medium, are going to germinate. And when the spores germinate, they can fuse, they can mate, and they can continue to proliferate, producing a new generation of cells. So if we can recapitulate these transitions, we will have every single of those networks. At least that's our hope. And my student Taylor, who is here at the conference, and my former postdoc Sandia, who is now a professor at Wesleyan University in Tennessee, they got this assay to work. And this is a time series that shows a single cell that starts arrested in a period of low CDK1 activity, and then it slowly progresses into making these little balls, which are spores. And then the spores are exposed to rich medium to trigger germination. Upon germination, the cells mate, and they change shape, and then they start to produce cells that are bigger than their ancestor cell. So as you can see, there's a lot of variation in shapes and size here, and we thought, well, we're gonna track the cells really easy, and actually it is not that easy. You look at the variety of shapes and morphologies that we have in these cultures is quite remarkable. Here we have this mosaic of cells, which shows different stages in the life cycle of yeast, and in order to track these cells, we had to tweak an algorithm that was originally developed for mammalian cells, for neurons. The algorithm is called CellPus, and the red heat map basically shows you the probability of a pixel being part of a cell. So if you see really red, that means that pixel is really part of a yeast cell. We had to train different models. So this is the general model, PROSEC. There is another one called SPOSEC, which specifically looks at the sporulating cells. We have one for mating cells, and we have another one that looks at the cells that are dividing. When we combine these segmentation models, which are based on convolutional neural networks, we can detect and classify all the cells according to a life cycle stages. So the assay looks like this in real life. Here you see the phase image with some cells, and they look red because they are cells. And this panel here shows you the probability of the cells being a spore. You will see that soon they light up in red. So the algorithm becomes able to detect the cells, but we don't see detections in the mating panel or the cell division panel because the cells are not there yet. They are just making spores, which are again these tiny little balls here. Now these cells will be exposed to germination through exposure to rich medium in a second, and you will see that this transition is quite fast. They're basically going to burst open, and we will lose the detection in the sporulating channel, and we will gain detections in the mating and the division channel right about now. So this is an algorithm that can take a look at every single life cycle stage of yeast. We thought, well, perfect, now this is going to be easy. However, the transitions sometimes are very fast, and this led us to a very interesting problem, which is almost philosophical. And that is, when the cells burst open so fast, how do you track them? When we look at things, in reality, we think that they exist in a continuum, in space-time, if you want. But what we recover at the microscope is usually a discontinuous time series, which is sparse. It doesn't contain every single thing that happened. And sometimes we miss events that are really important to track the cells or to understand what happened to the networks. And sometimes you cannot just go faster with the acquisition time because the cells are quite sensitive. So this poses a problem. How do we make up for the intermediate states that are missing? And my students, Shreya and Nika, they come up with a very bold solution for this. They just said, well, we have deep learning algorithms that are used by other companies like Disney, for example, to animate computer animated movies, and they can do very good frame interpolation. So you have two frames, you can use the information in the adjacent frames to synthesize roughly the image that was going in the middle. So we can create a synthetic depiction of the intermediate state. We can insert it in the real time series, and then we have this hybrid time series that should allow us to track the cells better. This is an example of how this looks. The real image is a time series at the frame rate that it was acquired at the microscope. Now the hybrid time series is going at a rate that will be equivalent of imaging every 15 seconds. And as you can see, in the real time series, there's going to be abrupt movements, but in the hybrid time series, the movements are going to be smoothened. And because they're smoother, it will be easier to track the cells. Now, this is quite amazing in terms of tracking, because now you have one cell in one position in the previous frame, and the cell moves completely to a different position in the next frame. You don't need to run an estimation of where this cell is that cell in the next frame. You could literally just segment the hybrid time series and track the cells just by looking at the mask overlap, other segmentations. As it is depicted here, these two masks, they don't overlap, so you will need a lot of complicated algorithms to figure out where this cell is that cell. But with interpolation, you can get this nice gradual movement that allows you to track the cells just by mask overlap, regardless of how they move. And we have a preprint release this year, which is now under review. In that paper, we are basically looking at organoids, immune system, mammalian cells, fungal organisms. It seems to be a very object-agnostic algorithm. Okay, so this is an example of the tracking under real conditions. This cell is going to germinate, and right now this is a real movie, and at this point we kicked in with the frame interpolation, so now it looks to slow down, and there's going to be a tiny cell that will emerge out of the germinating spore, and you will see how that cell is being tracked regardless of the changes in size or the changes in shape that it undergoes. We have now a lot of tracks, so we could actually model how this germination process looks like. It's quite interesting because it seems to be very stochastic when you look at it, but if you have tools to track the cells, there are actually some commonalities that have started to emerge. This algorithm, we think, can allow for tracking of the cells regardless of the morphology or the type of movement. Okay, so now we're going to be able to track the cells. What do we do now? We want to visualize the networks, and let's start just with this framework. A cheap solution for this that biochemists usually do is to take just one protein like We5 and another one like CLIP5, and then we're going to put a fluorophore or a protein that produces light attached to them, right? And these two proteins are going to be reporters. So by looking at them, we can infer the rest of the network. In the case of We5, it's rather easy because the protein is in the nucleus when the cells are in one configuration of the network, the low CDK1 state. And in the case of CLIP5 is in the nucleus in the opposite manner. Now, you're interested into how we measure this. We cannot use CLIP5 directly. We had to make a totally new sensor, which I would like to discuss with anybody that is interested afterwards. But the bottom line here is that if you see green in the nucleus of the cell, like here, that means that the cells are in a position when the configuration of the network is a high CDK1 state. And if you see red, that means that the cells are in a low CDK1 state. And this is going to be a movie of the entire life cycle of this organism. We're going to start with an ancestral cell, and this cell is going to undergo meiosis and all the different stages of meiosis, which are highlighted with yellow letters. And the plot has size in blue, the high CDK1 sensor in green, and the low CDK1 sensor in red. We see all the phases of meiosis, which is composed of two nuclear divisions. And once meiosis is done, the cells are going to make spores. The color of the area on the plot represents the cell that is being tracked. So we're approaching the point of germination, and we're going to see now this explosion that we can now track around this time. Now, there is a lot that we can gain from this time series. For example, in the published paper, was published in Molecular Biology of the Cell earlier this year, we showed that the size of the cells during sexual reproduction is dramatically larger than the size of the cells when they are undergoing mitosis or asexual reproduction. We also noticed that there is a decline in this transcriptional repressor, the red protein. It seems to decrease linearly up to the point that the cells trigger a reaccumulation of the green sensor. And there's a lot of things there which indicates that the cells are basically flipping between the low and the high CDK1 state. However, this is only 2 proteins, and we're using these 2 proteins to infer the whole network. So of course, that is not intellectually satisfying, because if you look at the network, it has already a lot of components. And sometimes we use terms like nutritional distress to refer to actually other networks. For example, the TOR pathway, which is a stress sensing pathway. And that pathway, we represented in a very simple manner, looks like this. And that is, already a lot of factors to take into consideration. So there is a technique in which you could increase the number of proteins that you could use to image the network. You apply the same principle. You take your protein and you attach it to a fluorophore. And this time we're going to take IME1, TOP1, GAP1, SFP1. If you make educated guesses of the proteins that you want, you can get to a point that you can image 6 different fluorophores. And I hope this move is going to be bright enough so that you can see that we have an hexagonal array over here, and we're going to be tracking 1 cell in yellow, and here we're going to plot the nuclear concentrations of that cell for 6 different proteins. These proteins have to do either with the CDK1 state controlling network or with the stress sensing networks. This approach can give us at least 6 time series per cell, and these cells are alive. So unlike transcriptomics data, we do have the previous state of the cell and the future state of the cell, so these time series contain a lot of information. And you can see, for example, that these proteins, even if you don't know anything about them, CDC10, W5, CLIP2, you can see that CDC10 behaves roughly like CLIP2. And when these 2 proteins, CLIP2 and CDC10, are high, W5 is down. And when W5 goes up, these 2 proteins go down. That is the textbook description of how this network should look like, because that's how the cell cycle networks are regulated. W5 opposes is the inhibitor at the top of the network that will repress the other proteins. Now, these 3 proteins, XBP1, STB3, and TOP1, are transcription factors that transmit the stress. And according to this data, they don't really care what's happening with the cell cycle. They are following their own schedule. So we hope that we can use now this time series to derive more causality between these proteins and start to make inferences about the network. And if somebody's interested in collaborating with us, trying to model this multidimensional, multivariate time series, I will be delighted to actually work on this, because we have a lot of them now. However, that was only 6 proteins, and networks in living cells can contain hundreds of proteins. And this is really a problem that we're going to have. How are we going to look at 100 proteins in a live cell without killing the cell and recording all that information? Well, I don't have the answer for that, but I would like to propose a trick to maybe get us there. We started to experiment with latent diffusion conditional image generation, specifically using a model called guided diffusion that was originally pioneered by OpenAI. And in this model, you can take a source image, for example, the image of the cells with the phase contrast microscopy, which is the cheapest one and is the healthiest one. And then you can train an algorithm to predict the fluorescent image without the microscope. So technically, you could train a latent diffusion algorithm to take the healthy way to depict the cells and produce predictions of how that image would look like had the image been taken at a fluorescent microscope. I hope that makes sense. So here we have 3 different predictions from 3 different diffusion models that use different type of algorithms like IS2B and DMT. And I hope you can appreciate that these signals, which are very bright in the target image, which is a real image, are recapitulated by some of the models. Some of the models hallucinate, like the third one. It has bright spots all over the place. But this seems to indicate that you could actually fine-tune these things to produce realistic, biorealistic fluorescent images from non-fluorescent images. So I guess you can guess what we're doing now. We are using our spectral imaging datasets that contain at least 6 different proteins per cell to train these diffusion algorithms. And at this point, we can get you at least 8 proteins from one image. So this is a single image in phase contrast microscopy. And all of these images are not real. They are predictions by latent diffusion models of histones, TON70, which is a mitochondrial protein, Rimilan, which is a protein that determines where the cells do spores or not, TPS1, a protein that controls carbohydrate metabolism, R6 controls lipid metabolism. This is a vacuolar protein, and this is our Wi-Fi transcription factor, and this is a transcription factor that is only expressed under stress. If we trigger the cells, you will see that the channels contain different information. And at this point, I can say that in some cells, actually, it's quite accurate. You could take the time series from the generated cell, and it's very hard to distinguish it from the real one. So hopefully this is going to be out soon, later this year. The paper is going to be called Generative Network Imaging, and it's going to propose the use of latent diffusion algorithms to try to extract as much network information from a single image as we can. With that, I would like to say that at least the answers that we have for those 2 questions that I said at the beginning are, you want to track the cells, you probably can use generative AI to increase the frame rate of the experiments. And if you want to track multiple things in living cells, maybe instead of shooting light at the cells, we could try to use the data that we already have and then try to come up with predictions, accurate biological predictions of where the proteins should be. Maybe like that we can get really, truly multidimensional data from single live cells. And with that, I would like to thank the laboratory that made this possible, and also the NSF-Simons National Institute for Theory of Mathematics Applied to Biology for inviting us here. I have to mention that the code from the previous papers is a pip installable Python package, so you can just Google it and you will find it. Right now we're at 30,000 downloads. We believe 25,000 of those are chatbots, but there's got to be at least 2,000 real people that were interested in this, and we're quite happy with that. As you can see, this was funded in part by the NITMB contributions. And if you want to know more about how we're trying to square off our spectral imaging, you can visit poster 34. Taylor will be glad to explain you the details. So thank you so much for your attention, and I'm happy to take questions.
Hi, great talk. When you were, I guess when you were developing the hybridization, where you would kind of fill in the time series steps, how did you evaluate those kind of, the hybrid images that the AI created?
Yeah, that's a really good question. How do we know that the images are accurate once we do the interpolation? There are different metrics to assess where images are accurately recapitulating the image that goes in the middle. I think in the paper let me check. We use something like this. So we can take a real time series and we downsample it. We remove the even number frames, for example. And then we take the odd number frames, ask the algorithm to produce the even number frame for which we have a ground truth, right? And then we can use a lot of similarity metrics to assess where the images are the same. For example, these are 4 generative algorithms that are trying to produce this original image. And these heat maps that you see here at the bottom shows dark spots when the pixels are different. So you can see that all of these algorithms basically produce images that are very similar, but if you look at the pixel-wise similarity indexes, they are very bad at looking at intracellular features. And I think that's why our tracking algorithm works, is because they're really good at doing the outline of the cell, but when you go into intracellular details, they're pretty bad. So that's a way to evaluate them.
Sorry, thank you for the talk. I was wondering, in both the applications that you showed, how would you deal with or kind of mitigate the hallucinations produced by the AI?
Yeah, well, hallucinations at this point can be brute force removed just by training with more data. But I think that if we really want to have images that are accurate to a point that you cannot distinguish them from, biological images, we probably need to invest in developing biologically informed neural networks that can bias these algorithms a little bit to produce things that are realistic. For example, we know that the network has to be in a certain state, so the protein should be in the nucleus, right? So if an algorithm will predict that the protein is outside the nucleus, it should be punished, right? So these are the kind of things that I think we need to develop in the future to reduce the amount of data that it takes to produce these results.
Hi, that was really cool. Thank you. Maybe this is a bit of a silly question, but what's the advantage of using a generative approach here instead of, say, like a more direct supervised learning sort of UNet approach? Like, do you need a generative step, or if you get your images in sort of pairs anyway, could you do it more directly?
Can you repeat the last part of the question, please?
So in your data collection, when you're getting your images, your fluorescent images, and also your bright field ones, do they come in pairs that, it's the same cells, right?
Yeah. The images for doing the translation from phase into fluorescence come in pairs. In our case, because we did the spectral imaging before, we have one phase image and we have 6 fluorescent images that pair with it. The only difference is the time that it takes for the microscope to move to the position that will record the next fluorescent channel, which is in the range of 200 milliseconds, 600 milliseconds, up to 1 second. So there's a tiny little gap, maybe of 200, 400 milliseconds between the images. But yeah, technically they should map very well one onto the other. I think you have a previous question before that, or?
Well, yeah. So if that is the case, then would it potentially be more data efficient to try and sort of train just a UNet to predict those fluorescence images directly? Or is there a proper mathematical advantage to using a generative approach?
Yeah, I think specifically for fluorescent microscopy, generative AI might have an edge because the noise in a fluorescent microscopy image usually is Poisson and normal. It's a combination of Poisson and normal distributions, and the signals are very weak most of the times. Because if you shoot a lot of light to the cells, they will experience phototoxicity. So if you have really strong signals, I think a CNN might do it, just a vanilla CNN. But if you're dealing with conditions where the signal-to-noise ratio is very low, you really need to record something out of that. I think that generative AI, specifically diffusion-driven processes, are really good at that.
Okay, that's really interesting. Okay, thank you so much.
Thank you.